Welcome to Medmarc's webinar series. Today's presentation is titled Revolutionizing Medicine, the Race to Unlock the Power of Gene Therapy and Build a Competitive Edge. I'm Kate Klaus, Senior Attorney in Medmarc's Risk Management Department. On behalf of Medmarc and today's presenters, Sheena, Wang, and Aydin Harston, thank you for joining us. Dr. Aydin Harston is a partner at Rothwell Figg. He integrates his scientific expertise into his legal practice to help his clients grow, protect, and defend the intellectual property that powers their businesses. Concentrating in the areas of pharmaceuticals, biotechnology, medical devices, health technology, chemicals, and mechanical devices and materials, he counsels and advocates for his clients in prosecuting patents and managing patent portfolios, as well as in the courtroom where he is an experienced litigator who handles every aspect of patent-related cases. Aydin has prosecuted over 1,000 patent applications, and he regularly handles IP diligence projects, participates in litigation teams representing clients in the federal district courts at the Court of Appeals for the Federal Circuit, and represents clients in inter partes review, appeal, and re-examination proceedings before the U.S. Patent and Trademark Office. Originally a scientist with a Ph.D. in biochemistry, he has devoted his career to studying and working with various aspects of science and technology. Based on his own research path and obtaining both novel and non-obvious results, he now focuses on helping inventors protect their discoveries. He devises creative and simple solutions and explanations for complex situations that enable clients to achieve their goals as efficiently as possible. Aydin is an editor of 2 firm blogs, RF Emerge, a resource for early-stage companies, and Biosimilars Law Bulletin, covering updates related to biologics and biosimilars under the Biologics Price Competition and Innovation Act. He has both a bachelor's and a Ph.D. in biochemistry from the University of Maryland College Park, and a J.D. with honors from the George Washington University Law School. Sheena Wang is an associate with Rothwell Figg. With a master's degree in biotechnology and a Bachelor of Science with a double major in genetics and cell biology, Sheena brings a background in both law and science to her role, integrating both in support of the firm's practice and clients. Her wide-reaching practice includes patent prosecution, patent litigation, client counseling, and trademark cases. In patent prosecution, she has drafted and filed patent applications in the biotechnology space, has experience building patent portfolios, and has conducted due diligence investigations covering FTO, patentability, landscape, company acquisitions, asset acquisitions, and investments. With respect to litigation, she has participated in litigation teams representing clients in the biotechnology industry and in trademark matters. Before joining the firm, Sheena worked on genetics research projects on topics including polymorphisms associated with various autoimmune diseases and genetic markers for use in plant classification, identification, and phylogenetic studies. She also has wide-ranging knowledge of the pharmaceutical industry through her work in various phases of the drug development process. These include vaccine analytical development and clinical trials from both laboratory and clinical settings. Sheena has a bachelor's in cell biology and genetics from the University of Western Ontario with honors, a master's in biotechnology from Johns Hopkins, and a JD from Washington and Lee University School of Law. And with that, I am pleased to turn things over to Aydin.
Great. Thank you, Kate, and the entire Medmarc team for organizing this and inviting us to speak. We're really excited today to speak about gene therapy and the technologies, the companies involved in this space that are doing groundbreaking work in revolutionizing medicine. And we're also going to talk about some intellectual property and patent aspects of this. So there's kind of 3 main categories that we're going to touch on. First of all, what is gene therapy? And how is gene therapy being advanced these days? The CRISPR-Cas system is something that we're going to talk about, and I'm sure the audience has heard about a lot in the press over the last several years. And as we are patent attorneys, we're going to talk about the patent landscape and considerations that go into that, and how the pioneers and the innovators in this field are growing their businesses through patents and also through the clinic. Because we are attorneys, we can't let you go until we give you some disclaimers. So these materials are solely for educational and informational purposes. The statements that we make today reflect only the personal views of myself and Sheena, and do not necessarily reflect the views of the firm Rothwell Figg. These materials are not legal advice, and they also are not investment advice. Each case and situation is fact-specific, and the appropriate solution will vary, and no attorney-client relationship is created as a result of this presentation. And with that, I'll turn it over to Sheena.
Hi, so I will be starting off the presentation today with a general background of gene editing. Going back to the beginning of human civilization, humans have been modifying the genes of plants and animals for generations. As an example, since we've domesticated animals, we've bred different kinds of dogs for hunting, foraging, and companionship. As another example, through years of selective breeding, many of the foods that we eat today, like bananas and cucumbers, look very different from their ancestral wild counterparts. And a slightly more modern example are genetically modified foods. For example, over 90% of canola available on the market today are genetically modified for herbicide resistance. Genetic editing technologies can even be used to resurrect foods. If you ever wondered what mammoths taste like, an Australian food company called Fox is working on cultivating mammoth meat in a lab to make mammoth meatballs. Also on the subject of mammoths, there's a company that is at the beginning stages on working to bring extinct animals back from extinction using CRISPR technology. One of its proposed projects is combining woolly mammoth DNA with the genes of an elephant, which is the mammoth's closest living genetic relative, to bring back the woolly mammoth. So, but before I get off on a tangent, and now that we have your attention, we will go back to the focus of our presentation today, which is using gene editing technologies for a new type of therapeutics. So starting with the question of what is gene therapy? Gene therapy is a technique that modifies a person's genes to treat or cure disease. Gene therapy therapeutics are being developed for a variety of diseases, including cancer, genetic diseases, and infectious diseases. The way these therapies work is either by using an ex vivo method called cell therapy, where cells are taken from the patient's own body or from a donor and are genetically modified before injecting the modified cells back into the patient's body. Or using an in vivo method, which works by injecting the therapeutic gene directly into the patient's body using a delivery method such as a viral vector or a lipid nanoparticle. Gene therapy works by replacing, inactivating, or adding a specific gene. The newly added gene will change the way proteins are created in the cell. So for example, if a gene is inactivated, then the protein it codes for will no longer be produced. Or if the patient's body does not create a functional copy of a specific protein, then gene therapy can be used to add a functional copy of the gene or replace the faulty copy with a functional copy. And then the patient's body can create normal functioning proteins. So this slide shows an overview of gene therapy drugs under development as of 2021. You're not expected to read everything on this slide. We included it to demonstrate that there's a lot of companies in this area, and this technology is applicable to a variety of disease indications. We wanted to use the slide to demonstrate that most of the therapeutics in this field are in its initial stages of development, and this is shown on the outside of this figure. And only a few companies have reached the center of the bullseye. And in the following slides, we're going to zoom into the center and look at the therapies that are on or nearly on the market. So zooming into the gene therapy drugs from the previous graphic that are the furthest along, Orchard Therapeutics has developed two therapeutics that's been approved by the EMA, which is the European Medicines Agency and the European equivalent to our FDA for the treatment of two inherited diseases. One is a condition that causes severe immunodeficiency, and the other is to treat a disease that affects the central and peripheral nervous system. Bluebird Bio is another company that has developed two therapeutics that's been approved by the FDA. for the treatment of two inherited diseases. One is for a type of inherited blood disorder, and the other is for the treatment of a rare progressive neurodegenerative disease. The idea of the idea of the slide is to provide a snapshot of the companies operating in this space. We noted the market cap and the number of employees in these companies, but as a note, these companies are constantly changing. So this number is really only accurate from when we pulled them, which is in June 2023. So continuing to look at gene therapy drugs that are the furthest along, another company with an FDA-approved drug is Novartis, which is a much bigger global biopharmaceutical company compared to some of the companies that we have discussed in the previous slide. So it shows that there's a big range of the type of companies that are working in this field. And it has developed a therapeutic that's been approved by the FDA for treatment of an inherited disease that causes progressive muscle wasting. Next is Roche, which is also a global biopharmaceutical company. It acquired a smaller startup called Spark Therapeutics, which developed a therapeutic that's been approved by the FDA for the treatment of an inherited disease that causes progressive deterioration of vision. Finally, we show GenSight Biologics, which has developed a therapeutic for the treatment of a rare mitochondrial disease. It previously applied for approval with the EMA, but has since withdrawn its application based on its indication, based on the EMA's indication that its current data is not sufficient yet to support market approval. So as a case study, let's look at Zynteglo, which is one of the recently approved gene therapy products that we've mentioned. Zynteglo is a treatment for beta thalassemia, which is a rare inherited disorder that causes a patient's body to make less hemoglobin than normal, which causes anemia. Particularly severe types of beta thalassemia happens when a person inherits two copies of the non-functional gene. Patients who have had severe beta thalassemia usually require frequent blood transfusions for the rest of their life. Zynteglo is a one-time treatment, and it's a type of ex vivo therapy, which means that it works by taking the patient's own blood stem cells, genetically modifying them in a lab using a lentiglobin viral vector to deliver a copy of the functional gene into the extracted stem cells. The modified stem cells are then infused back into the patient's body, which will start to make higher levels of hemoglobin. However, gene therapy treatments present some safety concerns. Some concerns are associated with the fact that the functional genes needed for gene therapy is often delivered using viruses, which are called viral vectors. Here, Zynteglo uses a lentiglobin viral vector. Some scientists, so while scientists have removed the disease-causing genes from these viruses, the body could still mount an immune response against the virus, which can cause side effects, including inflammation or even organ failure. Another risk is that viruses may affect the wrong cells, which can damage those cells. Another risk associated with gene therapy is if new genes get inserted into the wrong spot in a host DNA, which depending on the spot could lead to diseases such as cancer. Finally, a concern with using viruses as a delivery system is that the virus might recover its original ability to cause disease, and that could also cause disease in the patient that received the therapy. So for example, Zynteglo's label comes with an FDA mandated warning that there is a potential risk of blood cancer associated with this treatment. While no cases of cancer were seen in patients who participated in clinical trials for this drug, the FDA added a box warning because a few participants in another recently approved therapeutic, which works using a similar mechanism, developed a type of bone marrow cancer. So for the same reason, the FDA also required patients who receive this treatment to have their blood monitored for 15 years after they received the treatment. This slide shows a development timeline of Zynteglo based on its FDA filings and press releases. If you recall, on the previous slide, we showed all these companies on the outside of the bullseye that are at the beginning stages of development. So if you were wondering how long it takes to develop a gene therapy product, as you can see here, it took Bluebird Bio over 10 years to turn this from an idea to an FDA approved product. So the slide starts off with the submission of an investigational new drug application, which is submitted after the applicant has already obtained some core proof of concept scientific ideas. And that was in 2012. And this therapy was further developed over approximately 10 years and was recently approved by the FDA. So now we're going to move on to the next part of the presentation, which focuses more specifically on CRISPR as a gene editing system. Moving on, so as we've been discussing, recent scientific developments have paved the way for a new wave of gene therapies. CRISPR is the fastest, easiest, and cheapest of the gene editing tools in this new wave of gene editing science. And as scientists discovered in 1987, CRISPR is actually a naturally occurring process that functions as a bacterial immune system and defends bacteria against invading viruses. The naturally occurring CRISPR system is composed of two components. The first are short snippets of DNA sequences called clustered regularly interspaced short palindromic repeats. And the second are Cas proteins, which stands for CRISPR-associated proteins. When a virus invades a bacterium, the Cas proteins cut out a piece of the viral DNA through a series of steps, it copies it into an RNA. The RNA now acts as a guide. It binds to a Cas9 protein, and then the resulting complex searches for DNA sequences that match or more accurately complement the guide RNA sequence. So therefore, when the same virus invades again, the guide RNA Cas9 complex will recognize it and destroy the viral DNA by cutting it. In 2012, scientists were able to adapt the Cas9 guide RNA CRISPR system to target not only just viral DNA, but any DNA in almost any organism. And then later in 2013, scientists were able to show that this system can work in mammalian cells. So these and other pivotal discoveries show that the CRISPR system can be used as a precise gene editing tool that can alter DNA at specific positions and fix disease-causing genes, almost like fixing a typo. So this discovery eventually led to two scientists, Jennifer Doudna and Emmanuel Charpentier, to be awarded the Nobel Prize in Chemistry in 2020. So in a lab, the CRISPR system also has two components, the sequence targeting guide RNA and the DNA cutting Cas protein. Scientists can design a guide RNA which matches the gene that they want to edit and attach it to Cas. The guide RNA then directs Cas9 to the target DNA sequence in the host, and the Cas9 protein cuts the DNA on both strands on the precise spot. Once the DNA is cut, the host cell's own machinery will try to fix it. Usually the cell's proteins, called nucleases, will trim the broken ends and then join them back together. This process usually leads to the loss of some nucleotides, and that results in a gene that's turned off. However, scientists can also add a separate DNA template sequence to this CRISPR cocktail. In this scenario, cellular proteins can use the template DNA as a blueprint to repair the DNA break, which can repair a defective gene or insert a new functional gene. This ability to precisely edit the genome means that scientists can create treatments that target the underlying cause of genetic disorders like cystic fibrosis or sickle cell anemia, which previously can only be treated for its symptoms. And as we discussed in the very first slide, this system can also be applied to other organisms. For example, it can be used to increase crop yield or nutrition, bring species back from the extinction, or even as a research tool. So scientists can study what happens when a specific gene in an organism is turned off. So now I will turn this presentation over to Aydin, who will cover some companies specializing in CRISPR therapies.
Great, thank you, Sheena. On this slide, we're showing several companies that are at the forefront of pioneering CRISPR technologies. And you'll see that some of the familiar names from the timeline show up as being associated with these companies. So at the top, there are Intellia Therapeutics and Caribou Biosciences, which count Jennifer Doudna, who is a professor at University of California, Berkeley, as a founding member. And then below that, in the middle of the slide, is companies associated with Emmanuel Charpentier. That's CRISPR Therapeutics and ERS Genomics. And at the bottom, you see Editas Medicine and the Broad Institute, which are associated with Feng Zhang of MIT and the Broad Institute. And on the left-hand side of the slide, you see a lot of other excellent scientists that have contributed to the technologies that have given rise to these pioneering companies. And then on the right-hand side of the slide, we see some technologies that these companies have been working on, and this is by no way to be construed as a comprehensive list of the technologies that these companies are advancing. But just to give you kind of a flavor, CAR T cells, cancer therapeutics have been a major focus. Stem cells, muscular dystrophy, sickle cell, a lot of genetic disorders are targeted, and there's a lot of hope that these kinds of gene therapies can help cure these kinds of genetic disorders as well as cancers. And in addition, we see that there are also a lot of animal agriculture, plant research tools, and drug discovery aspects that can also be advanced by this type of technology and are being researched by these companies. On this next slide, we dig in a little deeper into these pioneering companies. So at the top, we talk about Intellia Therapeutics to give you kind of a flavor for their relative sizes. So we see it's about a $4 billion market capitalization, about 500 employees. Caribou is a smaller company, 137 employees, with a $275 million market cap. All these numbers are based on our research this month in June 2023. CRISPR Therapeutics is, again, larger company, 458 employees. And ERS Genomics is more of a licensing entity. So they hold over 100 patents worldwide covering the foundational intellectual property, or some of the foundational intellectual property surrounding gene editing and CRISPR technology. So they serve more as a licensing entity than a drug development company. And similarly, the Broad Institute holds a massive number of patents in this field and has done pioneering work for many years, and serves as more of a licensing entity and also a very advanced research institution. And Editas Medicine is another company that's producing and developing therapeutics, about 208 employees and a market cap of 600 million or so. On this slide, we listed some CRISPR therapy updates in 2023, some plans for clinical advancements. So we can see that a lot of the same companies that we were just mentioning on the previous slides are the ones that are making advances in 2023. So we see CRISPR Therapeutics, which is planning to submit its biologic licensing application by the end of this year. Editas is going to be updating its clinical trial progress. Intellia has a couple of products that are advancing through various stages of FDA applications. And again, CRISPR Therapeutics has a drug that will move into clinical trials. And I should have mentioned that CRISPR Therapeutics is working with Vertex Pharmaceuticals on the exa-cel product, which is nearing the bullseye that we kind of pointed to earlier. And in the middle, there's a company called Graphite Bio, which actually announced earlier this year that it was discontinuing its program for Nulacel and was going to focus on other gene therapies. So that Nulacel update is a negative update, I guess. I'll hand it back to Sheena.
All right, so in the next few slides, we're going to look at patent portfolios of some of the companies associated with Doudna, Charpentier, and Zhang as representative examples of the patent portfolio of companies focusing in the CRISPR space. So starting with Intellia Therapeutics, which is based in Cambridge, Massachusetts. Intellia was co-founded by Doudna and Charpentier in 2014 to develop CRISPR-based therapies. Our search results show that Intellia has been filing patent applications with the US Patent Office and the WIPO, which is the World Intellectual Property Organization, starting in 2016. So according to our search, 5 of those applications are currently been granted into US patents. The blue indicates pending applications, which includes both US patent applications and pending PCT applications being examined by the US Patent Office and the WIPO. PCT applications indicate that Intellia is protecting its intellectual property worldwide. A PCT application is filed with the WIPO and can be designated to enter national stage in any of its 157 contracting states within 30 to 31 months after filing. So therefore, after a PCT patent application is designated for national entry, it becomes multiple patent applications that are being examined and issued by the national patent offices of member countries. So I should note that we focus the search on US and WIPO patents and patent applications. So you are not seeing the national stage entries outside of the US. And also as a disclaimer, this graphic and other patent search graphics of this presentation reflect only the number and legal status of patents and patent applications at the time we conducted the search. So therefore, they only represent a snapshot in time. This slide shows Caribou Biosciences' patent portfolio. Caribou was co-founded by Doudna in 2011 and is based in Berkeley, California. It is focused primarily on using CRISPR technology to develop treatments for cancer. Our results show that Caribou has been filing patent applications with the US Patent Office and the WIPO beginning in 2012 and has over 60 granted US patents. CRISPR Therapeutics was co-founded in 2012 by Charpentier and is based in Switzerland. Our search results show that CRISPR Therapeutics has been filing patent applications in the US Patent Office and WIPO beginning in 2016, and it has over 50 granted US patents. So Editas was founded in 2013, and its co-founders included Zhang and Doudna, although Doudna has since left the company. Our search results show that Editas has been filing patent applications with the US Patent Office and the WIPO beginning in 2014 and has over 20 granted US patents. Finally, this slide shows the patent portfolio of the Eli and Edith Broad Institute of MIT and Harvard, which was formally launched in 2014. As we kind of touched on before, the Institute's research goals are not only to research CRISPR technology, but to allow scientists to organize collaborative and interdisciplinary projects to work on different challenges in molecular medicine in general. Therefore, search results are a little over-inclusive on this slide because it includes the Broad Institute's entire patent portfolio, which covers more than just the patents directed towards CRISPR technologies. This slide shows 15 organizations with the highest number of CRISPR patents and patent applications as defined by our search, which includes patents and patent applications using the word CRISPR, gene editing, guide RNA, or Cas, narrowed by some classification codes from the International Patent Classification, or IPC, that are directed towards human therapeutics. Overall, we can see that the organizations associated with Doudna, Zhang, and Charpentier, such as the University of California, Berkeley, Harvard, MIT, the Broad Institute, Intellia, Caribou, CRISPR Therapeutics, and Editas are among the organizations with some of the biggest CRISPR portfolios. Also notably, while ERS Genomics is not represented here, as we discussed before, it's a licensing entity. And so some of the patents that would be assigned to, for example, the University of California based on Doudna and Charpentier's work are represented in this graph, even though you can't see ERS Genomics here. So another search we conducted was looking for patents that specifically have independent claims, which are claims that are meant to cover the core of the invention directed towards guide RNA. The results of this search show that many of the organizations conducting research directed towards CRISPR technology in general, as we showed in the previous slide, are also conducting research directed towards components of the CRISPR system, such as the guide RNA components. So for example, we can see that Intellia, CRISPR Therapeutics, and Editas are all represented in the graph showing the top 10 organizations with the biggest patent portfolios directed specifically towards guide RNA. So the CRISPR system will only work within a cell if it's properly delivered into the designated host cell. Therefore, we designed a search to see which organizations are filing patent applications covering one of the possible delivery systems for CRISPR technology. This search focused on finding patents and patent applications directed towards human therapeutics and mentioned within the patent that it can be used for CRISPR or gene therapy, and then also have independent claims directed towards inventive lipid nanoparticles or components of lipid nanoparticles. Lipid nanoparticles are also called LNPs, and they're one way to deliver the CRISPR-Cas complex into a cell. So interestingly, the organizations filing patents in this field are less linked to the companies associated with Doudna, Charpentier, and Zhang that we've been looking at compared to the guide RNA search. So this could be because LNP technologies can be used to deliver other therapeutics. And then additionally, this could be because companies focusing on CRISPR therapeutics would prefer to concentrate their funding and research efforts on its core technology, and then will plan to either start developing their delivery technologies later on or just licensing it from another company. Out of the organizations that we discussed in our previous slides, notably the Broad Institute, MIT, and Intellia, appeared to be developing LNP delivery technology along with their CRISPR technology.
All right, so on this slide and the following slides, we're going to talk about the top challenges in the CRISPR field. And I think the first part of that is, as you can kind of see from the bullseye map and the number of patents, it's getting to be a crowded field. So, you know, based on and you get different numbers depending on how you search these things, but depending on the search, based on the search that we ran, we found that there are more than 13,000 pending patent applications and patents globally on CRISPR-related technologies. And as the fields advance and get more crowded, there are more publications and and patents, the anticipation and obviousness issues increase, as do also concerns for double patenting, where overlapping inventions are claimed, repeatedly, which is prohibited by different types of laws in different jurisdictions. Other than that, safety issues are another important challenge in this field. So various studies and clinical trials have been put on hold by the FDA and other regulatory agencies for safety concerns. We saw kind of an example of that with a few of the companies that we talked about earlier today, like Graphite Bio. And the risk of off-target activity is a major safety concern here, where the therapeutics may not only impact the intended site of action, but also act on other sites and continue to cause mutations and genetic changes, and that leads to genomic instability and can also disrupt the functionality of otherwise normal genes. And as we've seen, the costs of developing and administering these gene therapies are very high. So we saw a number earlier about $0.8 million per treatment, although it is for a cure. So as an estimated cost for one of these therapies. The stocks of these publicly traded biotech soared to all-time highs during the COVID pandemic. There was a massive amount of investment into biotech and pharmaceutical companies. And since then, those stock prices have tumbled. And this is partially due to clinical regulatory setbacks that have happened, and also the rising funding costs and the economic climate have also caused these impacts. Sheena, was there anything else on the slide that you wanted to talk about?
Nope, I will continue to, I'll pass it to the next slide.
Great, thanks. Yeah, so on these slides, we're going to talk about the top 5 considerations for a successful patent application or patent portfolio plan in this area. So if, when an inventor or company is coming in and considering the patent process, and they have an idea or they have an invention or they have research, first of all, we want to understand what is the state of the art? What have others already patented? What have others already published about? What other products are out on the market or in development? How close is that to what the inventor is interested in doing? Then once we understand that, of course, we want to know what are the differences. What are the novel aspects of the invention? Would those differences have been obvious or not by a person of ordinary skill who's comparing the invention being developed to what exists in the published literature and in the field? After that, we want to understand, do we have enough experimental data, particularly in these kinds of fields where there's a lot of challenges, a lot of unpredictabilities, a lot of failures, a lot of skepticism. The patent examiners are going to require experimental data to really support the claimed invention beyond just written disclosures or kind of theoretical or thought experiments. So, in considering how much experimental data is already available, that'll inform you, about timing of filing a patent application. You don't want to file a patent application too early where your data is insufficient, because that will not pass the thresholds of providing sufficient written description and enablement for a person of ordinary skill to make and use the claimed invention and to recognize that the inventor actually had possessed the claimed invention. And another thought is, are you coming in and trying to claim too broadly? do the experimental data and disclosures you have really enable one to make and use the full scope of what you consider to be your invention, or are you just asking for much, much more than you've actually demonstrated or reasonably enabled? It's kind of a double-edged sword though, because you can't wait too long and complete all the experiments, because it is a first inventor to file system. So it is a kind of a race to get to the patent office. So if someone else files before you do, and you are waiting for that last experiment to finish up, that inventor that files first will get the patent. Another consideration in the patent space is how far along is the invention. So patent exclusivity lasts for 20 years from the filing date. So a patent filer will want to take that into consideration. We've seen that there's long development times, and we've also seen that it's a crowded field and a race. So there's a balancing act and a lot of competing factors to take into account when deciding when to file, because if you file too soon, and your patent term will be diminished by the time you actually have a commercial product. If you file later, others may file before you and get the patent, and or others may publish research that can be used to reject your claims as being not novel or not obvious. The other part of it is the licensing revenues. What aspects can be patented and licensed to others potentially? A lot of these platform technologies are kick off considerable licensing revenue. So if someone can get a patent on that and at the right time to be able to license it out, that can be the best outcome. And then as the technologies advance, the filer would want to consider seeking additional patent protection on various aspects and improvements as time goes on. And this will, newer filings will have later expiration dates. They'll build the strength of the portfolio. They'll build the revenue streams for licensing. So the inventor should not rest on their laurels and should always be thinking, what else am I doing? What else can I think about? And how can I continue to build my assets? Patents are assets. And finally, you want to avoid creating prior art against your future patent filings by disclosing just kind of early ideas. A lot of inventors have many brainstorm sessions, come up with lots of ideas and want to dump them all into their first patent application, and without having sufficient data or sufficient detail, and that can really work against you, because that will become published, and then later 5-10 years down the line, once the invention's actually kind of fully baked, that prior art will come back and be used as a rejection. So it's something to be avoided. Other considerations are, what is your exit strategy? Different companies have different business models, and one could be out licensing, another could be developing a product and selling the asset or the company. Also, joint ventures with a partner are very common in this kind of space, where a larger partner that has experience will come in and jointly develop a product. Or finally, company can go it alone and try to take it from beginning to commercialization through the regulatory process. Finally, the key markets are an important consideration. What is the global patent strategy? Which countries will you need patent protection? What are the key markets for sales? And also production, manufacturing is also important. There are different types of protection that can be sought in those countries. So that, Patent Council can, of course, advise on the best types of protection to seek. And then finally, you have to determine what the budget will be and the costs of filing and protection and seeking product approvals in all those markets, and what will be the kind of envisioned payback of those costs incurred.
Hey, well, we hope you enjoyed our webinar. Aydin and I are excited to answer any questions. that you might have, or if you have any questions that might arise later, feel free to reach out to either of us. We provided our emails here, and also Medmarc has our contact information, and we'll be happy to continue this discussion with you. So now we will open it up to Q&A.
Thank you both so much. This was really very helpful. We do have a few questions for you. First, if a company wanted to file patents throughout the R&D process, what are the main stages of development that they should be thinking about as milestones in filing these new applications?
Sure. So I think that in any development, let's talk about this field in particular. So there are various aspects that we've talked about that go into this kind of technology. There are delivery methods, there are payloads, which are, like the guide RNA and the Cas proteins. And there are going to be manufacturing processes. The later stages will become more therapeutic in focus, the treatment indications, the patient populations. And then after that, there are even further stages as the clinical trial process goes through, different kinds of reactions and side effects and optimizations of dosing and dosing regimens will come up. So I think each of those phases is a fertile ground. So you want to start at the very beginning, talking to your experienced IP counsel who's been through this process kind of from A to Z. And get advice rather than either rushing off to file a patent application too early or waiting too long. Oftentimes, these developments come out of research institutions that have time pressures to publish or partners that want to publish, and that can really create problems for when a poster presentation or a webinar gets out that discloses the invention before there's been a patent application filing. So it's really important to start early, engage with counsel, and work collaboratively, keep counsel in the loop so that you don't miss anything. You don't let any assets kind of slip through.
Thank you. What are some concerns about obtaining patent licenses in the gene editing space?
So I think we've seen a lot of issues in this, especially in the gene editing field, with the pioneers in this field that have filed various patent applications, obtained different rights, and have also been litigating with each other in various countries around the world. And those litigations continue to rage, and there are other players also involved besides those that we've mentioned. So we get this question a lot from companies that are asking, what licenses do I need? Who do I need a license from? And there are a lot of complexities and nuances there, because there is kind of a lot of overlapping IP and a lot of cases that are in various stages of being litigated or appealed. And so depending on how those cases turn out in various countries, sometimes you get conflicting results in the US versus Europe or various jurisdictions. One licensing entity may end up with the patent rights and another may end up losing the patent rights. So it's quite complex to determine who to actually get a license from. And so that's, again, a situation to be explored in great detail with counsel to determine what the needs of the company are, what the timing is. Can you wait until there's more certainty in this field, or do you really need to, for whatever reason, have some kind of license now, and then make those decisions and negotiations with a very well-informed viewpoint?
Thank you. And another question, we work with a number of companies that are in the emerging market space. They might be startups, they might be developing new technology, new products. And by necessity, they have to do a lot of collaborative work. They might be working with researchers. They might be trying to seek funding from investors. They might be looking at contract manufacturing partners or critical vendors. And this collaboration really necessitates disclosure of some information about their products. What are the best ways for them to protect their IP as they go through these discussions?
Yeah, this is a point that comes up extremely often. There's always a baseline. People say, let's sign a confidentiality and non-disclosure agreement. And it's often boilerplate that gets presented whenever two companies want to discuss a potential deal. And my advice would be to not merely rely on NDAs as it's very risky actually to rely on NDAs. So there should be other procedures put in place in addition to that. Again, this is a really important area to engage with counsel early. Don't just have the business folks go off and sign various NDAs and other agreements without having them vetted. There's often terms in those agreements that are not undesirable and can be improved. And also there's other steps that we can take before those discussions take place. One of which is to file provisional patent applications. This will show that you've filed a patent office, you have your idea of your invention and some details, and you have done that before receiving any confidential information from the other party or before disclosing it. There's a lot of data breaches recently. So with any party that you enter into, you have to consider the risk. What if there's a data breach? What if hackers obtain this and just publish it or do other nefarious things with my confidential information? What then? So it's always good to have a provisional patent application at least filed on whatever confidential information is being disclosed. And then another thing that I advise is to make these discussions more in general terms for as long as you can. So you may have excellent results and talking to a funder, and the funder may not necessarily care about the specific details of what your molecule is, your specific delivery vehicle formulation, or other such detailed aspects. They want to see that you've got something that really works. So you can show them the data without telling them the actual inventive aspects, which would be those guide RNA sequences, LNP formulations, other kinds of delivery aspects, and various other much more specific scientific details that are going to form the claimed invention, but that funder or that partner may not necessarily need to know that information to make a decision whether or not they want to do this deal.
And just to jump in really quickly, I don't believe we've talked about provisional applications. So those are there are applications that you can file, for example, with the U.S. Patent Office, and they are not published, and they are they'll stay kind of in a black box with the Patent Office for a year, and then you can choose to convert it to a non-provisional application, which will be examined and then eventually, hopefully, issued into a patent application, or sorry, into a patent.
Thank you both very much. This has been really an interesting topic. It's incredibly complex, and we appreciate your ability to break it down for us. So Aydin and Sheena, thanks so much for your time today.
Thank you.
Thanks.
It's been a pleasure.